Okay, so for this unit, we'll be looking at T cell and B cell development and activation. For this audio lecture, we'll focus on Chapter 8 and looking at T cell development. So you'll remember from an earlier chapter that T cells develop in the thymus, and so the earlier T cell precursors are going to develop in the bone marrow, and then they're going to traffic to the thymus. And then once they enter the thymus, they're going to be in the cortex towards the outside, but closer to the medulla because that's where the blood vessels are going to be located. So if you look in that figure, you can see how the medulla is towards the middle of the structure. And then as you move out, you move into the cortex. So again, these T cell precursors are going to move towards the cortex, towards the outside, and then what they'll do is then migrate back into the medulla when they have undergone selection. So if we look at this more closely, we can see how at that cortex medullary junction, how we have the blood vessel present. And so this, again, is where our precursor cells are going to come in because they're coming in, they've left the bone marrow and come to the thymus to develop. You'll notice that as it moves from the cortex medullary junction through the cortex and then into the subcapular cortex, it's going to go through these different stages, these different subsets. So they're denoted as DN1, DN2, DN3, and then as it comes back into the cortex, DN4. The DN refers to double negative. These cells at all of these stages have no expression of CD4 or CD8. Okay. But the stages of 1, 2, 3, and 4 refer to expression of c-KIT, which is a stem cell marker, CD44, which is for adhesion, and CD25, which is the alpha chain of the IL-2 receptor. And we'll talk more about those and what the levels look like for those different stages, but just keep in mind that for those 3 different markers, the expression level would be different for each of these subsets. So DN1 versus DN2 versus DN3 versus DN4. Okay. So again, as the cell is in that cortex medullary junction, again, we're having shifts in expression of these different markers so that it would then become a DN2 that moves it into the cortex. At this point, that cell has now committed to the T cell lineage. So prior to that, at stage DN1 precursor and also DN2, it is possible for the cell to give rise to myeloid and natural killer cells. But by the time it's entered in to the subcapsular cortex, they can't go back. So it's committed to the T cell lineage. So by the time when it is DN3, that is when the T cell gene rearrangement's going to occur. And so if you remember back to the last unit, we talked about how the T cell receptor is made up of 2 chains, and there's 4 different possibilities. So we have alpha, beta, gamma, and delta. And so we can get different T cell arrangements, but we also have the gene recombination. So we have different gene segments that can be produced in our T cell receptor. So all of that rearrangement is occurring during that. To kind of gain re-entry into the cortex, we then have our 4th stage, or DN4. Okay. At this point during the transition between in the cortex, one of the major events is the expression of CD4 and CD8. So we'll see how you can see how the cell is now DP, or double positive. So it's going to express both CD4 and CD8 initially, and then it will become single positive so that they would either express CD4 or CD8. So it's during positive and negative selection stages that the cell will select which type of cell it's going to be, what type of T cell it's going to be, whether it would be a CD4 single positive cell or a CD8 single positive cell. Keep in mind when we're talking about the term double positive or single positive, that's just referring, or double negative, it's just referring to CD4 and CD8, that there's a slew of other markers, just like we were saying about c-KIT, about CD44 and CD25, but there's also additional markers like CD3 that are present. So just because it is double negative or double positive or single positive, it doesn't mean it doesn't have expression of other surface markers. So one of the important aspects of development of the T cells in the thymus is that, this is where we're going to be screening to remove any autoreactive cells, because after this, after it goes through that migration through the cortex and back into the medulla, it can enter into the bloodstream. And the idea of that is so that it can go and move into secondary lymphatic tissue and get activated and see pathogens, see damage signals, right? But it should not be seeing healthy self. And so we do want to make sure that any of those are removed. And so this is where that recombination of the T cell receptor through gene segments is really important. And that this is happening during the double negative stage, so that we can either get the combination where we have alpha beta or gamma delta T cell arrangements, T cell receptor arrangements. And we do want to keep in mind that when the cell first arrives into the thymus, they're not technically T cells, so they haven't undergone that differentiation. They still have the potential to become natural killer cells, dendritic cells, B cells, and myeloid cells. That's actually really important because remember that these cells are going to undergo selection, and part of that selection means that we need to have presenters, right? And so this ability to become other cells is important, again, because then it brings presenters into the thymus to present to the T cells, but it also means that if we have excess amount of T cells and we don't need to produce more, we can halt that, right? So Notch is the receptor that's known to be expressed and commits to the T cell lineage. So we can see here in this figure how if we have Notch positive cells and we feed growth factors, we'll end up with a T cell lineage. However, we take these same cells and they're Notch negative, feed them the same growth factors, they do not become T cells, they actually go to the B cell lineage. Okay, so again, Notch becomes very important in order to commit to the T cell lineage. And this is in part because of the GAT3 transcription factor becomes activated when Notch is binding to a ligand. So as I mentioned, these cells are initially in a double negative stage, and so generally we would call these thymocytes. Okay, so these are cells in the thymus. At each stage, as I mentioned, there's going to be changes in the expression of several different molecules. The molecules that are generally looked at to characterize at what stage they are is c-KIT. So I mentioned this is related to the stem cell, and so this is the receptor for stem cell growth factor. CD44 is an adhesion molecule, and CD25 is the alpha chain of the IL-2 receptor. And so, again, as we mentioned, the T cell receptor rearrangement, it's going to begin at the double negative 2 stage, but most of the rearrangement is going to happen during the double negative 3 stage. So looking at the different expression of the double negative thymocytes, you can see how double negative 1, this is the stage it's at when it goes from the bone marrow into the thymus. And so this is going to have high expression of c-Kit because it's going to be the closest to the stem cell stage. We also have CD44 expression. Again, that's important because we need to be adhering and moving through. Okay. When we look at the double negative 2, so DN2, we still have expression of c-Kit, expression of of CD44, but also CD25. So we're beginning to express receptors for IL-2, so we can start to sense IL-2, and that can affect the development of the T cell. Okay, so this is going to be in the subcapular cortex space, and we're going to begin that. We've got the T cell lineage commitment, and we're going to begin rearrangement. So selecting for the expression of the different gene segments. For DN3, this is where we're no longer expressing our adhesions, so we have CD44 is negative, but we've decreased our c-Kit expression, and we have CD25 expression. This is when we're going to get the pre-T cell receptor expression, and we're also going to undergo beta selection. For DN4, at this point now we've further down-regulated c-Kit so that it's low or to no expression, and we've also turned off expression of CD44 and CD25 because we've gone through the process of selecting for the T cell receptor, so this starts to signal for the T cell to go into the cortex, and then eventually with the focus of becoming a double positive thymocyte. Okay, so any additional rearrangement that needs to be done of the receptor for beta and alpha chain would also occur during this stage. So keep in mind for the receptor, for the T cell receptor, we are going to have either alpha beta or gamma delta. Okay, so with T cell receptor beta arrangements, this is the one that's going to first take place, the one that's more likely to be productive. So if it's going to be more likely to be productive, so let's try that first and see if that works before you switch over. So when we look at T cell receptors, they're going to be more likely to be alpha beta than they are going to be gamma delta. And so gamma delta is actually more common in fetal development, and if we think about fetal environment, that's going to be different. So there's going to be different signal cues, but as the organism develops, that gamma delta arrangement becomes less common. Okay, so for our purposes, we're going to focus more so on alpha beta T cell receptors. Okay, so looking at our progression through development, again, as we've mentioned, we started this double negative 1 subset, and then we're going to progress through double negative 2 to double negative 3. Again, changes in expression levels, where we go from expressing CD44 to not expressing, and then we go from not expressing CD25 to expressing. So those go inversely in their expression levels. So that in DN3 we have our pre-T cell receptor, and then we go through a beta selection. So we want to make sure that we have a good beta chain for that. This allows us to enter into DN4. Okay, this is before we have expression of our double positive. Okay, now we're going to focus on the alpha chain, so any kind of rearrangements of the alpha chain can be made. At this point, after we've both had the beta selection and we've had alpha chain rearrangement, that T cell receptor is considered good. And so now we can expend the energy to express CD4 and CD8. And because this is a good T cell receptor, we're going to express both so that we could go, because that cell can divide, right? And it could become CD4 positive or it could become CD8 positive, okay? So that receptor is undergoes negative selection. So with negative selection, we're making sure that it's not going to react or bind to any of our self-peptides, okay? So if it fails negative selection, it would undergo apoptosis, okay? So if we pass negative selection, we can undergo positive selection, where it will then become a single positive, so it would either be a CD4 or a CD8, okay? You can get further selection of different effector function, so this is where you could get your T regulatory cells, your T helper cells, your cytotoxic T cells. So again, early in the thymocytes development, those double negative thymocytes are going to undergo the beta selection. And keep in mind that each point of this, after it passes a stage, we can have proliferation and then further differentiation occurring. So after beta selection is an area where we would have proliferation occurring. Because again, if that beta chain is good, then we don't want to have to go back and go through beta selection again, go through this whole process again, because at the next round we're now undergoing the alpha chain rearrangement, and so that means that even though a single cell underwent beta selection, it now has a possibility to undergo different types of alpha chain rearrangement. So it gives you a bigger pool of cells to go through. So looking at positive and negative selection, again, double positive thymocytes are going to make up the majority of the cells because, again, you've just undergone proliferation with beta selection. You're going to undergo another round of proliferation after alpha rearrangement. And so the majority of your thymocytes are actually going to be these double positives, and so they need to undergo selection. So for positive selection, it's going to select thymocytes that have receptors that are capable of binding to MHC, to the MHC molecules that are presented. And they need to have that with a low affinity, okay? And so positive selection in that it can bind to the MHCs in which this individual produces, right? With negative selection, this is where we want to make sure that any of these receptors that bind at high affinity are eliminated, okay? Because if they're going to bind at high affinity, they could end up binding self, okay? And so we don't want those in circulation, right? And so this is one of the steps that allows us to build tolerance. Tolerance being that we're not going to respond to self, okay? So most cells will actually fail positive selection, and so 95% of them end up failing, which means only 5% end up surviving. Within that 5%, some of those are still autoreactive. And so, again, with negative selection, this helps to eliminate some of those. And then there's other steps once out in circulation that also help to eliminate the negative effects of autoreactive T cells. Okay, but again, if a cell is responding incorrectly, they can end up going through apoptosis. And again, for positive selection, you want it to be able to bind to MHCs, but at a low affinity. And so if it's positively selected for, it would move through. If that affinity is too high, then it would be removed. And again, that would be called negative selection. And at that point, now the cells can become single positive. Okay, so this graphic just shows us how in the cortex we are going to have our double positive thymocytes. So they would be expressing CD8 and also CD4. They would have their T cell receptor. They would have some of the co-stimulatory molecules also like CD3 present, along with other surface markers also. So any cells that are responding with a high affinity would undergo negative selection, so they would be removed. They would undergo apoptosis. If they have no interactions, which is a possibility, so they're not able to bind to the MHCs, so they would end up undergoing death by neglect. Okay, and then what we're looking for something is low to intermediate affinity, and that would then be positively select for. Okay, because again, these ones can bind with the MHC, but it doesn't bind so strongly that it would kind of artificially activate, short circuit, if you will. Okay, so once those positive selected thymocytes that are double positive undergo this, they can then become single positive. And again, they're going to undergo proliferation before that, so some of those cells would become CD4 positive and stop expression of CD8, or other ones would be CD8 positive T cells and stop expression of CD4. Okay. Once in the medulla, they're going to undergo presentation with self-antigens. Okay. And so this is another round in order to determine whether there's any autoreactive T cells. And so now we have a round of negative selection happening. So again, you don't, as a T cell, you don't want to respond to self. And so any that do are deleted out. These cells are now allowed to go out into circulation and considered mature T cells. So I would just say that, the caveats here is that, some T cell receptors aren't going to be able to bind. So again, those ones are going to undergo neglect because they're not going to have any kind of stimulation because they aren't able to bind to those MHCs. Okay, then you have the ones that are going to bind too strongly. So you want to remove those ones because again, they could kind of artificially activate. So those undergo negative selection and apoptosis induced. You're looking for the Goldilocks. You're looking for the one that binds just right, not too strong, but still able to bind. Okay, and so those undergo positive selection, so they become single positives. So what we're looking at is MHC restriction, right? So it's really about that T cell receptor being able to bind to the MHC, okay? And again, hitting that Goldilocks where we're getting the right amount of binding. And so if we look at this figure, we can see that the ones that actually undergo positive selection are going to be only 2 to 5% of those thymocytes that begin this, right? Begin this selection process. That the majority of them are going to fail to develop and be selected for, so they're going to die through neglect. Other ones are going to bind too strongly, and so they will be negatively selected for and induce apoptosis. And then some of them do survive, but they're going to end up becoming T regulatory cells. So they'll actually help regulate the immune response. Okay. And as I was mentioned, negative selection is going to ensure self-tolerance, and negative selection is what's referred to as central tolerance. So it's at this point where we have um, you know, the most ability before these cells go out into circulation to eliminate any of these autoreactive cells. So with that, because we've deleted the cell, because they've been induced to go into apoptosis, this is sometimes referred to as clonal deletion induction. So inducing apoptosis to delete the clone, so we can't make any more copies of it. So we do want to keep in mind, as I mentioned, not all thymocytes that recognize self are deleted for. So there are ones that are able to kind of escape. And so the reasons for this is because not all tissue types are expressed in the thymus. So the thymus is expressing all the, proteins that it has in its genome, but there can be variations of that, right? And so it's not going to express every single protein that could be expressed in the body. It expresses most of them. And so there's little variations, especially ones that have derived during the development of the organism, those that aren't being expressed in the thymus, that is a potential for autoreactive T cells. So autoimmune regulator is an important protein in this process because it actually causes the thymus to produce proteins so that they can then be processed and presented on MHCs. Okay, so there are mouse models that have mutations or deletions of AIR, and so they are used to study this process. So again, keep in mind with natural selection, there are also other mechanisms that play a role in self-tolerance, and so we can get clonal arrest. So this is where autoreactive T cells are prevented from actually proliferating and dividing and maturing further. We can get clonal anergic. So this is where autoreactive T cells are inactivated. So they're not deleted, but they're kind of, it's like they're put to bed. They're hibernating. So they're deactivated in a sense, usually through either signals to the receptors or through the production of different cytokines. And we also can get clonal editing. So this is where the T cell gets additional chances to edit their T cell receptor. So this is where adding or removing nucleotides from the T cell receptor genes can give you a different receptor that binds differently and maybe no longer binds something that's autoreactive. This is with the alpha gene. And then we also get clonal deletion. Again, kind of last resort, where you delete the clone out so that it's not able to go out in circulation. So, why don't we delete all the cells that are positively selected against? So even if we have the same T cell receptor, we can get different affinities. And again, this has to do with some of the editing that happens. This is known as the affinity model. And so the different strengths of binding ends up giving a different signal. And this is really critical in whether the T cell is allowed to develop or not. And so they've used mouse models that have a single T cell receptor, so all the T cell receptors are the same, so they only can recognize one peptide. But because of the MHCs, class 1 molecules that are expressed, that we can get these different affinities, and so this then sends a different signal to the T cell so that you get a different selection that can happen. So looking at this figure, you can, this kind of elucidates what I was just talking about. So, if we have no peptide present, there's not going to be that ability for the T cell receptor and for the APC that has the MHC to actually interact, and so we get death by neglect. we can have the peptide present, but because of the MHC that's present, we would actually end up having a low affinity to it, right? And so that's ideal, so that one gets to survive and go forward. We could have a different MHC, and then that would cause a high affinity, and then we would end up getting a negative selection or death. There's also the alternative model, and this looks more at the peptide that's involved, so sometimes referred to as the altered peptide model. So keep in mind that peptides produced by the thymus, these are coming from those epithelials, are coming from the neighboring epithelial cells, are unique, and they can be different than the peptides that are going to be generated from other cells. And so again, what's being selected for is what's in the thymus, and that's not always an indicator of what's going on in the whole body. So we do want to keep in mind that it's not necessarily the affinity model or the peptide model. It's maybe somewhere in the middle, and so they both need additional research going on to kind of elucidate which model maybe is the more dominant model that's happening. So do positive and negative selections occur at the same stage in development or in sequence? So it's most likely that negative selection actually occurs at various points in development, and that positive selected cells, they have to express CCR7, which is a chemokine receptor, in order to move into the medulla for further development and selection and screening. So the situation is likely complex, and so that it's not necessarily linear as it was presented, and that the process isn't necessarily in a certain sequence. And again, this is where additional research and understanding of the process and utilizing different mouse models and such may be able to elucidate more on this. So there are several models that have been proposed to explain lineage commitment. So how we end up progressing to that single positive T cell. And so one of the models is the instructive model, which is in panel A. So in this one, the T cell receptor in CD4 versus the T cell receptor in CD8 have co-engagement and have end up generating unique signals. Those signals that are generated instruct the T cell which lineage to fully commit to. And so they will, depending upon those unique signals, it would end up yielding a different single positive cell. The stochastic model, this looks at the idea that one of them is going to be randomly downregulated. So either CD4 gets downregulated or CD8 gets downregulated. And so it's more of a flipping of a coin in which lineage it would end up going in. And that if the correct receptor isn't, if the correct co-receptors aren't actually expressed, then that induces the cell to go into apoptosis. So remember, we have these co-receptors that also have to be expressed. And so if you have randomly selected this CD4, but you're expressing the co-receptors for CD8, then you would go into apoptosis, as such as the inverse would go. Okay. The kinetic signaling model states that cells commit to a CD4 lineage if they receive a continuous signal, but if they do not receive the continuous signal, they will become a CD8 lineage. So CD8 lineage has an interrupted signal, in that you have IL-7 that's actually going to promote CD8s to differentiate. Okay. Um, and so, again, if we look at the instructive model, it's that signaling is going to instruct the cell what lineage to come in. Stochastic model, it's a flip of the coin. And the kinetic signaling model is one that incorporates in somewhat both of those, in that there are certain signals that will say become a CD4, and then the default is if those signals aren't constant, if there's a kind of break in that signal, then the default is that you become a CD8. Okay. So this just kind of outlines what I just said. And then there's just a slide for each one, so I won't dwell on it. But again, just keep in with instructive that we have signals that are going to occur because we have MHC T cell receptor activation, and that generates signals that then tell it to become CD4 or CD8. With this stochastic model, it's just a flip of the coin randomly. We are going to downregulate CD4, so that means we're going to have CD8 expression. Inversely, we're randomly going to downregulate CD8, so we're going to have high expression of CD4. And then with kinetic, again, it's about duration, if you will, of the signal. So if it's a constant signal, it's going to say CD4, 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 CD4. But if there's a break in that, the cell defaults into the CD8 lineage. So double positive thymocytes can also become other types of thymocytes besides T cells and B cells. And so we can get natural killer T cells, so slightly different than your natural killer cells that did originate from the lymphoid progenitor. So these ones will actually express as T cell receptor, but they'll have an invariant alpha chain, so it hasn't gone through the same selection process. These usually interact with the CD1 molecule, and this is what's used, if you remember, for lipid antigens. Okay, we also have intraepithelial lymphocytes. These are usually have CD8 expression, but also have some of the features of innate immune cells. So they're not, they don't have the same specific type of reaction that we would see with like a CD8 positive T cell. And then we also have our regulatory T cells. So these are ones that are going to be from the CD4 subsets, and they help to regulate adaptive immunity. Okay, so these different outcomes all are going to rely upon what signal cues were given to the double positive thymocytes. Traditionally, the more common pathway would be single positive, either CD4 or CD8, and going into the helper and cytotoxic lineages. So these single positive T cells will then exit the thymus and undergo final maturation. So there's a number of cascades of events that's going to happen for this final maturation stage. So one of those is FOXO1 expression. It's a transcription factor, so that gets upregulated, and that causes upregulation of a number of other receptors. Again, keeping in mind that these receptors allow for those single positive T cells to sense what's going on around them. So S1PR is one of the critical receptors that needs to be present in order to allow the T cell to exit. So cells that have this mutated or deleted aren't able to actually exit the thymus. Also, IL-7 receptor is also going to be upregulated, and CCR7, which is a chemokine receptor, that also gets upregulated. So this is going to help the cell to survive as it exits and moves into the lymph nodes. So once the T cells have just exited the thymus, they're known as recent thymic emergence, or RTEs. Okay. They're not technically functional mature yet as older cells because they haven't undergone activations and such. So there's additional, you know, changes to some of those surface expressions uh, when you compare to uh, T cells that are in the lymph nodes or have left lymph nodes. So just to talk a little bit more about self-tolerance, keep in mind with the T regulatory cells, they're going to be critical in maintaining self-tolerance because again, keep in mind some of these cells might have escaped even though they're autoreactive, that might not have been picked up in the thymus. And so these T regulatory cells are going to continue to help maintain self-tolerance. So some of the functions of the T regulatory cells are going to be deplete the local area of stimulating cytokines. So they're going to bind those cytokines, so they're not going to be available for your other T cells. They can also produce inhibitory cytokines so that they send a signal for those cells not to activate. They can inhibit APC activity, so that those APCs aren't able to or don't present to other T cells. And they can also directly kill T cells. So if these other methods, aren't working, if you have a T cell that is overreacting, that T regulatory cell can induce the cell to go into death. Okay. And one of the markers that's used to look at a T regulatory cell is FOXP3 expression. So we do want to keep in mind that there are also peripheral mechanisms of tolerance to make sure that these autoreactive thymocytes don't damage tissue. Doesn't always happen, but we do want to try to eliminate that from happening. So some self-antigens are hidden, and so which means that APCs lack the costimulatory molecules needed to actually initiate an immune response. So these APCs aren't able to recognize them, so they're not able to engulf them from their source. Okay. In some cases, these self-antigens are presented by non-APCs, and that actually will end up preventing the initiation of autoimmunity. And then also if we have strong self-antigen signaling through the T cell receptor, but in the absence of co-stimulatory, so we have these, we'll talk about in our next chapter, we'll talk about how we have to have these co-stimulatory molecules present in order to activate the T cells. And so in some cases, those co-stimulatory molecules don't get involved, and so that actually initiates the T cell to go into anergic mode, which is that they're non-responsive. So just to summarize, the big key pieces that you should take home from this chapter is that the development of the T cells, the thymocytes, arise from multipotent CD4 negative, CD8 negative, so double negative precursors that are going to migrate from the bone marrow into the thymus. That these mature T thymocytes have a diverse T cell repertoire because they've undergone the beta chain selection and then the alpha chain rearrangement, so that they would be able to sense presentation off of the MHCs. That the fate of each of these thymocytes really depends upon the affinity of the T cell receptor to the peptide and to the MHC complex. So that will dictate whether it's able to survive or not. There are a number of mechanisms that are in place in order to help remove autoreactive T cells during development, which is central tolerance and that this is further reinforced and maintained in the peripheral to make sure that we minimize the chance of autoreactive T cells. That both the CD4 and CD8 thymocytes have to survive positive and negative selection to be allowed to migrate through the thymus, and that all these steps are highly regulated. So, regulation from what cytokines are being produced, what surface markers they have, so what receptors they have, so that they can sense those different factors too. Okay, so there will be a kind of second part to this, looking at chapter 10, which is going to be the activation of T cells, and then there will also be two additional audio lectures for B cells with their development and activation.